Hey guys, I'm Melena.
I'm Nora.
And I'm Lily.
And
we're the PROTOZOA sisters.
And in this video, we're going to be going over signal transduction. To start, let's talk about your basic signal transduction pathway. First, the ligand binds to a receptor protein located on the plasma membrane. Once the receptor is activated by the ligand, it triggers an intracellular signal. This signal is then amplified through relay proteins such as secondary messengers and kinases, leading to a response in the cell.
Now let's talk about G-protein-linked receptors. G-protein-linked receptors are a type of cell surface receptor that receive a wide range of extracellular signals, including hormones and neurotransmitters. In this process, a ligand will bind to a corresponding receptor, triggering a conformational change of the G protein, causing it to activate, releasing GDP and binding to GTP. The activated G protein subunit dissociates and interacts with other proteins, initiating a signaling cascade in the cell. This leads to various cell responses.
Now I'm going to talk about receptor tyrosine kinases, which are a class of enzyme-linked receptors that play crucial roles in various cellular processes, including cell growth, differentiation, and survival. They are activated by a variety of growth hormones and factors. First, we will add two ligands onto the extracellular domains of the receptors. These two receptors will join together and form what is called a dimer. This process is called dimerization. Then, we will add 6 ATP, 1 ATP for each of the tyrosines. This puts a phosphate group on each of the tyrosines and leaves us with 6 ADP. This process is called autophosphorylation. This is when the dimerized receptors phosphorylate each other on specific tyrosine residues in their intracellular domains. This will cause the recruitment of signaling proteins, which is phosphorylated tyrosine residues that serve as docking sites for various signaling proteins such as adapter proteins and enzymes. Then they will be activated, which will cause a cellular response. The signaling proteins initiate a cascade of intracellular signaling events. This leads to things like gene expression, cell proliferation, and cell differentiation.
Next, let's talk about ligand-gated ion channels. When a ligand binds to the channel, it causes a conformational change, opening the channel and allowing ions to pass across the membrane, altering the concentration gradient and triggering a cell response. After this is accomplished, the ligand then leaves the channel, causing it to close again.
Next, let's talk about the epinephrine pathway. Epinephrine, also known as adrenaline, is a hormone and neurotransmitter that plays a crucial role in the body's fight or flight response. In this process, epinephrine binds to G protein-coupled receptors on the cell surface. This kicks off a chain reaction. Once it binds, the G protein splits into two parts, alpha and beta gamma subunits. The alpha subunit goes on to activate an enzyme called adenylyl cyclase, which turns ATP into CAMP, or CAMP. CAMP is a second messenger that powers up protein kinase A, which then gets to work by adding phosphate groups to various target proteins, triggering a phosphorylation cascade that eventually leads to the inhibition of glycogen synthesis and the promotion of glycogen breakdown.
Now, a cascade is a series of chemical reactions in which one molecule activates multiple other molecules, amplifying the cell's response. Let's look at an example. A phosphorylation cascade begins when a ligand or other signaling molecule binds to a receptor that is on the cell membrane. Once binded, this then sends a signal to an active relay protein that turns an inactive kinase into an active kinase. This active kinase, shown in pink, then phosphorylates another inactive kinase, shown in blue, producing ADP. This process then occurs once more, creating an amplified cellular response.
Now let's talk about CAMP, or cyclic adenosine monophosphate. It is a crucial second messenger molecule involved in many cellular processes and acts as a messenger within the cells, relaying signals from hormones and neurotransmitters. CAMP works when a hormone binds to a specific receptor on the cell surface. This binding activates a G protein, which then activates the enzyme adenylyl cyclase. This activation converts ATP into CAMP. CAMP activates protein kinase A, pKa, and then this pKa phosphorylates various proteins, leading to a variety of cellular responses such as changes in gene expression, cell growth, or metabolism. You may be thinking that these two look pretty similar, but in CAMP there is a second bond onto the phosphate group.
And that's it for this video. Thank you guys so much for watching. Make sure to like and subscribe to the Protozoa Sisters channel for more AP Bio videos.
Did somebody say slave?